Today's lecture is going to be on genetic therapy, stem cells, and cloning. What I want to present to you today is the promise and perils of gene therapy and stem cell research. I'm also going to point out, so we're going to focus on the science purely, but I also want to point out where if someone does have an issue with research done in one of these areas, I want to point out where the most common points of concern are. I'm definitely not suggesting that you have one opinion versus another, but I just want to point out where issues lie if someone does have an issue with one of these lines of research. But really, we're going to focus on the science today. Okay, so remember, whenever we do gene therapy, there's many ways to deliver gene therapy. I want to focus on the plasmid again today. We really focused on the plasmid in the last lecture, so just make sure you can review these components. You know what antibiotic resistant genes are, you know what the origin is, you know what LAC-Z does, what palindromes are, things of that nature. Okay, again, shows the process, right? Again, just make sure you're familiar with this process. Okay, so actually in my my graduate work, I actually explored some questions to try to use nanotechnology to target genes, and I want to sort of illustrate one of these examples now to you today. The example I'm illustrating to you, the goal was to use titanium dioxide nanoparticles to deliver gene therapy to cancer cells. But the example I'm going to show you today is more of a proof of principle, just saying, could this possibly have worked? So what we have here is we have a nanoparticle called titanium dioxide nanoparticle, indicated by this gray circle. Tube 4 here is our experimental group. 1 through 3 are our controls. So I'm going to focus on 4. But notice that 1 through 3 are all lacking something that 4 contains. Okay, so we have our nanoparticles here. We chose titanium dioxide because they're very photoreactive, so we could use them for therapy later on if we wanted to. We've explored that in the lab a little bit. We also chose titanium dioxide because they're considered very non-toxic to humans. They're found in many, many commercial products, toothpaste, sunscreen, the paint that you put on your walls, like the shiny paint, the semi-gloss. Because they're very non-toxic unless you activate them. So we thought it was a good nanoparticle to use. Then what we did was we coated the nanoparticle with a fluorescent dye so we could see it. So this red fluorescent dye is just coating the nanoparticle so we can see it in these experiments. Then what we did was we took something called a peptide nucleic acid. It's a DNA analog, so it's like DNA. It has same basis as DNA, but a modified backbone, and that modified backbone allows a PNA, or a peptide nucleic acid, to hybridize with a DNA even more strongly than a DNA could hybridize with a DNA. So this black strand here is something called a peptide nucleic acid. So we designed this sort of probe, if you will. Okay, here's the other half of the experiment. Then we took a plasmid DNA, right, a piece of plasmid, and we cloned our gene of interest in it, the gene that we wanted to deliver to the cancer cell to deliver some sort of therapy. And so you can see here that that gene of interest is encoded by this blue strand right here, right, by this blue strand. That's our gene of interest. And so we designed it in a way that our PNA, or our peptide nucleic acid, is complementary to our gene of interest. And we wanted to use these nanoparticles to deliver this plasmid containing the gene of interest into a cancer cell. That was our goal. The first thing we did, though, was before we did that, we wanted to show that we could actually use the nanoparticle with the PNA, hybridized to the gene of interest, to move the plasmid around a test tube. So this is purely in a test tube. And what we knew was if we took these samples, we mixed them, and if we centrifuged them, we knew that nanoparticles would pellet to the bottom of the test tube, but DNA under the same conditions would not pellet. Okay, that was something that we knew beforehand. But keep this in mind. The DNA would not pellet unless it was somehow anchored to the nanoparticle. In tube 4, you could see it's anchored through the hydrogen bonds between the PNA and the target gene. Tube 3, tube 2, and tube 1 are all missing something, right? So there's no target gene in 3, there's no blue insert, there's no PNA in 2, and then 1 has no nano anything, right? So there's nothing there. Okay, so then what we did was we incubated the tubes, we spun them at a very low centrifugation speed, like 0.2 G, and then what we did was, this is the expected result, right? So we looked at them, we thought we'd get something like this, where the plasmid would only come down in tube 4. That's what we thought we would get. This is what we got. When we took the pellets, we ran them on a gel. This is the same experiment repeated 3 times, okay, 3 independent replicates. you can see that in tube 4, this row here is our DNA that we're acquiring. This is just a ladder to show the size of the DNA. You can see in tube 4 in each situation we get a large amount of DNA pulled down. Tube 1 we get nothing, right? We don't see any detectable DNA in tube 1, so that's holding true so far. 2 and 3 we see something, right? So there's obviously some non-specific interaction happening in this experiment, but needless to say, it's a lot less than in tube 4. So we took this as a good sign. We ran some stats. The stats appeared to be significantly different, right? So tube 4, we're pulling down more DNA than the other control tubes. That was our proof of principle. We could sort of move it around. Then what we did was this. We introduced the contents of tube 4 to a dish containing human breast cancer cells. So these are our human breast cancer cells growing in this dish. And we wanted to see, would the nanoparticle and the plasmid be taken up together in the same cancer cells within this dish? And one thing I want to mention about the previous slide is the plasmid itself that we used, when you put it into a eukaryotic cell, it would express a green fluorescent protein. Now, we didn't have any protein in mind exactly, right? It was sort of a scrambled sequence, but we'd see green in the cell, just again, a proof of principle. And so what we did was this. So you could see that if we look at this, we took a picture using a confocal fluorescent microscope. Every field of view you see is the exact same field, so the camera's not moving around the slide at all, or the slide's not moving underneath the camera, but rather what it is instead of having taking the whole picture, so here we have like a whole picture, right? This image is called a phase contrast image, but instead of seeing that, what we see is we put different filters in front of the microscope each time we snap a picture. So we put a filter in so we'd only see the green, a filter so we'd only see the red, and then these are overlays, which I'll explain in a second. So what we saw is, if you focus on the center cell, right, here's the cytoplasmic membrane, here's the nuclear membrane, here's the nucleoli, we see that the center cell is lighting up red, indicating that it received our nanoconjugate. Also, the center cell is lighting up green, showing that it probably received our plasmid, right, because it's producing a green fluorescent protein. That was a very positive sign. Now, I'm saying it's the middle cell, but we have to overlay, right? So we have to overlap these images here to truly show it's the same cell. And you could see that if we overlap red and green, we get yellow. There's a little yellow tinge here. So yeah, it is the same middle cell, and this complete overlay shows us it is. So that was a positive sign, a way to perhaps use nanotechnology to deliver gene therapy. Now, I abandoned this experiment, and the reason I did is because you could see that only 1 out of the, about 10 cells in this image actually received the plasmid and the nanoparticle. So the transfection efficiency, transfection being introduction of DNA into a eukaryotic cell, the transfection efficiency was very low. So we got, a positive result, but it was low efficiency. So we sort of abandoned the experiment, but at least shown you an attempt to use nanotechnology to deliver gene therapy. What I'd like to do is I'd like you to watch these different clips here. So there's 2 clips I want you to watch, and I have 3 links because one of the links always has trouble working, but there's 3 links. The one link that says mouse study is showing you research that individuals have done at Northwestern University with stem cell research. And then this other link, this one or this one here, you have to try them both, but shows you the personal side. So it shows you about a scientist who changed his research in response to his daughter being paralyzed. So he was always a neurologist, but he went into more stem cell research to regenerate spinal cords to help his daughter who had been paralyzed in a skiing accident. And this is not his daughter over here, but this is a researcher in his lab. So I'd like you to watch these videos so you can see the personal side of stem cell research and then see the scientific side, see where spinal cord regeneration was in animals at the point that this video was made. Okay, but let's talk about stem cells too, so you could understand the genetic potential of cells and better understand that video that you'll watch once this lecture is done. So there's 2 types of cells. There's differentiated cells and there's stem cells. So differentiated cells are cells that all contain a complete genome, but they already have their purpose, right? So they're a skin cell, or they're a liver cell, or they're a heart cell. They've already been differentiated. In other words, they already have a specialized role. They don't have much genetic potential. Now, I have this little caveat here. I probably should put an asterisk here instead of a little tick mark, but some individuals are trying to reprogram differentiated cells. So they're saying that if we can reprogram, they have the potential to express all the organism's genes and produce any cell. But again, a huge if here, right? If differentiation can be reversed or reprogrammed. And there's some belief that that could be possible. So, but if you're not reprogramming, these differentiated cells are already specialized. They lost their potential. On the other hand, we have stem cells. Now, there's two types. There's embryonic stem cells and adult stem cells. Now, all stem cells have the potential to become different cells. Right? That's where the potential comes in. Now, there's different degrees to which they have this potential, so we'll talk about that in a second. But what I mentioned here is embryonic stem cells have the highest level of potential. They're called totipotent. Once they lose a little potential, we call them pluripotent. Adult stem cells only are multipotent. So they have potential, but not the potential to change into a huge set of diverse cells that stem cells do. We'll talk about that in more detail in a second here. Okay, but why are some researchers trying to differentiate cells that are already differentiated? Like, why would they do that? Well, one reason is if you could take a skin cell, for example, and differentiate it, if you could unprogram it and make it a stem cell again, then you have an unlimited supplies of cells. Right? There's no shortage of skin cells. If we can turn them back into stem cells and then redifferentiate them into what we want to, we have an unlimited supply of cells, and it avoids any concerns that individuals might have if we're using embryos in the research. Now, what makes us think that that's reality as opposed to science fiction? Well, other organisms actually can do this themselves, right? We see it a lot in the plant world. If you take a few cells from a carrot root, you could see that we can actually, those cells, if you put them in the proper conditions, they can produce an entire adult plant again. So this unprogramming of cells, or this reprogramming, is something that nature can do. Now, humans can't do that, right? But perhaps in the biotech setting, we can make that possible. There's some individuals actually from the University of Wisconsin-Madison who have shown that they can reprogram skin cells. And there's other papers that have come out recently as well. Different procedures to do that. One of the most recent papers saying that by soaking cells in amyl acid you can reprogram them actually has not been replicated to my knowledge yet. So I don't know if that's universally accepted, that that's really a viable technique. But it's a line of research that people are pursuing. Okay, so let's back up again and talk about stem cells. Let's return to that topic. So stem cells have three properties, right? And really I'd almost consider two properties, and two are very closely related. So some people say three, but it might be better to think of them in two. So the first one is they have the ability to self-renew, right? Stem cells can produce more stem cells. They also have the ability to divide asymmetrically. And these two I would link together. I think they're almost one bullet, really, but sometimes people separate them to two. What I mean by this is if you can imagine a stem cell here, when it divides into two daughter cells, one of those daughter cells will stay a stem cell. The other one will start to differentiate to become a specialized cell. So that's why on the left, the stem cell can renew, and on the right, it's producing a specialized cell. And the fact that one side is different than the other, the two sides are different, the two daughter cells are different, that's this asymmetric cell division. Okay, another property of stem cells is the ability to relocate and differentiate. So stem cells can literally migrate around the body if they have the correct platform or scaffold to move upon. Okay, let's look at embryonic development a little bit. Just sort of a crash course. So you have an egg and sperm that unite, you get fertilization. That first cell is called a zygote. Then what happens after that is the cell divides to the 8-cell stage. It's called a morula. In Greek, morula means cluster of grapes. I think that might be ancient Greek, I'm not sure, but it means cluster of grapes. And at this stage, this is where we get the totipotent stem cells. That's where we get them. So they can turn into anything in the body. Then, once we get to this next stage, this blastocyst, we have cells that are called the inner cell mass. And these cells are already differentiated a little bit, right? So they're still stem cells, but they're to the point where they're only pluripotent. So they lost some of their potency. On the next slide, I'll show you exactly what they lost. Then, basically what happens is we have implantation of the embryo, and we get a fetus eventually, right? If that's what our final result is. Let's talk about in vitro fertilization. So it works through that method, right, in a sense. Sperm and egg are united in a dish. They're grown up and then they're implanted into a woman to see if it would take and produce a viable offspring. It's a very interesting procedure. So there's many embryos that result from this procedure. And here's where an issue comes in. So I'm not saying it is an issue, but if someone does have an issue with stem cell research, this is typically where they would, or at least one of the areas they would. And so the idea is this. There's many embryos that are produced. Some of them are implanted into a woman, right, to see if a pregnancy will result. But the other ones are frozen for later use. Or if they're not needed, they're frozen indefinitely. Or if they're not needed, they might be frozen and then used for biological research. So that's where the issue is if someone does have an issue. And again, I'm not saying there is an issue, but just if someone did, that's where it lies. So it's something to think about, right? Think about your feelings about that topic. Okay, again, another slide showing you this, but just illustrating some points a little more specifically. So at the beginning stage, at that morula stage, we have the totipotent stem cells. They can literally form an entire human being in theory, right? I'm not saying it's being done right now, but they could in a biotech sense. And so what I mean is they can generate every cell in the body, including, and here's the key word, the placenta and extra embryonic tissues that are needed for pregnancy. Once you get down to the inner cell mass of the blastocyst, they're pluripotent because they cannot form an entire human. They can generate every cell in the body, but they cannot generate the placenta or the extra embryonic tissues. So that's the potency they lost at that stage. Once it differentiates further, you have cells that are called hematopoietic stem cells or neural stem cells or mesenchymal stem cells. These cells here are more multipotent. They have lost some of their plasticity. Plasticity is almost a synonym for the word potency, honestly. So saying, like, what could they turn into? And so hematopoietic stem cells can turn to different things, but they can only turn to different types of blood cells. They can't turn into nerve cells, right? Contrarily, nerve cells or neural stem cells can differentiate into different types of nerve cells, but they can't go into stem cells. You sort of get the idea. So at that stage, we have cells that are considered multipotent. And here, again, these cells here, the ones that are not differentiating, can stay at stem cells, and we can, culture stem cells indefinitely. Okay, just a little concept check. Maybe print that slide out, see if you can fill in those gaps. Okay, so there's two different types of stem cells, as I mentioned. Let's focus on this a little bit more now. So there's adult stem cells, there's embryonic stem cells. Embryonic stem cells, most people will believe, are the most promising because they're totipotent, but adult stem cells are also pursued. There's adult stem cells in the bone marrow, for example, and all of these stem cells are being pursued for different types of clinical applications. So, for example, to cure paralysis or diabetes or Alzheimer's. So there's many different routes that people are pursuing. I'm not saying that we've cured those or made the progress we want to make yet with our stem cell research, but there's a lot of promise in these different areas. Okay, adult stem cells are stem cells that help you heal, right? Think of it that way. So you're not generating, all the different variety of cells that you could with embryonic stem cells, but rather, as I mentioned before, hematopoietic stem cells give rise to different blood cells, right, et cetera, et cetera. There are also stem cells I want to mention here that are found in the amniotic fluid, and these might be more flexible than adult stem cells. There's also stem cells in the umbilical cord. When a child's born, something you'll encounter, we've already encountered if you have children. If you're going to have children, you'll encounter it. Many facilities are saying they'll freeze down the umbilical cord to save those stem cells for your child for later on in life to see if there's some benefit for them later on. I'm not saying you should do that or should not do it, but it's something that you want to take some time and think about. Okay, again, this is just a diagram showing you the difference between adult and embryonic stem cells and what they can turn into and what they cannot turn into. So we've covered this already, but it's a nice little visual. This is that umbilical blood cord banking that I discussed with you earlier. There's a lot of companies out there. The most famous one is called Viacord. They will find you if you are pregnant and then you're going to give birth. I'm not sure if they get a hold of doctor databases. I don't know, but they will find you. And what they do is they say, for a service, we'll freeze down the stem cells in the umbilical cord or the placenta, and we'll save them for your child for later use. Again, not saying you should do it or should not do it, but something to think about. There's obviously a cost associated with that if you're going to keep it for personal use. So, something to think about. Stem cells can also be used for cloning. And so in 1997, Dolly the sheep was cloned, right? And the technique used to clone organisms like this, or one of the most popular techniques, is called nuclear transplantation. And so what happens is you have an egg cell, the nucleus is removed with a microinjector or a microneedle, and what happens is a donor cell nucleus is added there. So in other words, basically a somatic cell nucleus from whatever adult you want to clone is added to that egg. Then you have that egg that's grown up, that fertilized egg that's grown up, and eventually you have an embryo. At this stage, you have to decide, do you want to implant it into a surrogate mother who's primed for pregnancy? She's primed with hormones. She's also primed perhaps by mating her with a sterile male, just to create that act. But with the male sterile, so he's not the one impregnating her, but rather it's this embryo that's implanted. So you could do that to go ahead and clone an individual like Dolly the sheep, or you could take these stem cells from this embryo and grow them in a dish for therapeutic purposes. So I show this with the cloning of animals, right? But this is, again, where if someone has an issue with stem cell research, this is often where they do, because the same model is not exactly, but at this stage, the same model is applied to humans. So either it's implanted in a human woman to give birth to a child, or it's kept in a dish and it's frozen or kept in a dish and it's used later on for different types of therapeutic research. So again, that's sort of where the issue could lie if someone did have an issue with the technique. Students always ask me, are we cloning humans? To my knowledge, no. Many countries have a ban on that, actually. In 2001, there was a little bit of, excitement / hysteria about a biotech company that announced that they had cloned the first human embryo, but the company had said that it was proprietary knowledge, in other words, secretive knowledge, and they couldn't reveal how they did it. So I think most people assume that that was not a real incident, but more just sort of publicity. But nevertheless, something to think about. Would you be okay with the cloning of humans? That's something that eventually, could be possible. Like I said, many countries ban that right now. But if the technology did exist, is it something you'd support or not? Something to think about. Again, I don't have the answers, but it's good to raise the question. Again, please watch this video. There's two of them. There's one about the mouse research, and there's one on the personal side. I'd like you to watch these videos, again, just to bring the issue a little